Okay, so let's go ahead and get started. The general layout of the nervous system, we're looking at the central nervous system being the brain and the spinal cord, and the peripheral nervous system being everything outside of the brain and spinal cord. So if you keep that in mind, that the brain and spinal cord is the central nervous system, and then everything else is going to be the peripheral nervous system. Peripheral nervous system is then broken up into two parts, and you'll see in a diagram that I'll provide in just a minute that the nervous system is all in sets of two. Makes it real easy for us to study that way. But the peripheral nervous system broken into somatic and then autonomic. So the somatic nervous system, and we'll go over this a few times also, you'll see soma in a neuron, you'll see somatosensory, you'll see somatic. Soma means body. That's what it means. So soma in a neuron is the neural body, or the neuron body. Somatosensory is sensations from the body. Okay, so somatic nervous system is going to be the bodily nervous system. This is the nervous system that controls our muscles and so on. Afferent nerves are sensory in nature, and efferent nerves are motor in nature. Efferent, if you think efferent and exit, that means that these nerves are leaving the central nervous system. Afferent nerves are those that are coming towards the central nervous system. Okay? So that makes sense that efferent nerves run from the central nervous system out to your muscles and so on in the body for motor movement. And then the sensations from your feet or your legs or your arms are coming towards the central nervous system, therefore through afferent nerves. The autonomic nervous system. This is the nervous system that is just that. It is automatic. We don't have to think about it. We breathe, our diaphragm moves, our heart beats without conscious effort. And then it is broken up into two groups also, the sympathetic and the parasympathetic nerves. We'll talk about those in more detail here. Okay. In the autonomic nervous system, all the nerves are efferent. So again, efferent means that they're exiting the nervous system. The sympathetic and parasympathetic nerves generally have opposite effects. So for the sympathetic nervous system, we're going to talk a lot about the fight or flight response. So the sympathetic nervous system gets us aroused, okay, gets us prepared and ready for action of some sort, and the parasympathetic nervous system then calms us down. Okay, so if one were to increase heart rate, that would be the sympathetic. If it were to decrease our heart rate, it would be the parasympathetic. Two-stage neural paths. The neuron exiting the CNS synapses on a second-stage neuron before the target organ. Not too important for you, but just to know that it's a two-stage neural pathway, and we'll talk about, we haven't even introduced neurons yet, but that'll make more sense when you realize that some neurons have very, very long axons, and some of those neurons then connect to other neurons that have very short neurons or axons, or they're interneurons where they really don't have axons at all. Okay, so here again, the autonomic nervous system, see sympathetic, you see the fight or flight there. Parasympathetic, you see the rest and restoration, and that's the important part here. For the sympathetic, the second stage neurons are far from the target organ, whereas in the parasympathetic, the second stage neurons are near the target organ. Okay. And then here's that diagram that I was mentioning to you. So you have the nervous system broken up into two groups: central nervous system, peripheral nervous system. Central nervous system, two groups: brain and spinal cord. Remember, and as I mentioned, everything aside from the brain and the spinal cord is going to be the peripheral nervous system. So let's move over to that one. Now it's broken up into two again: somatic nervous system, soma, body, bodily nervous system for motor movement, etc. Autonomic nervous system, automatic for our heart rate. Okay, and in both of these we have afferent, efferent nerves. And we went over that with exiting the nervous system. Efferent, exit the nervous system. Afferent, come move towards the nervous system. Okay, and then under the autonomic nervous system, we also have sympathetic nervous system and parasympathetic nervous system. The fight or flight with sympathetic, the rest and restoration with the parasympathetic. And so that works for our major division. Okay, all right, let's move forward with our meninges, ventricles, and cerebral spinal fluid, CSF. The CNS is encased in bone. Of course, everybody realizes that you have the skull, okay? But not a lot of people know about the three meninges. There's the dura mater, which is a really tough outer membrane, the arachnoid membrane, which is web-like, and guess what? Arachnoid, arachnids, spider, yes, there's the connection, web-like. Pia mater is actually adhered to the cortex of the brain. If you were to try to pull the pia mater off, you would actually destroy brain tissue. It's stuck on there. It's kind of like saran wrap stuck onto a food of some sort. And then you have the cerebral spinal fluid, which helps cushion the brain. And we'll go into more detail as we see here. Here's our diagram. You have your lateral ventricles. The lateral ventricles are filled with cerebral spinal fluid, and then from the lateral ventricles they flow from the third ventricle to the fourth ventricle using the cerebral aqueduct, and then it actually flows down into the spinal cord through the central canal, and you can see all of those in figure 3.3. Here's a little in relation to the cushioning effect. So you have the scalp, then you have the skull, very, very hard bone, skull. Then you have the dura mater, the arachnoid mater, or here it's called meninx, but meninx meaning that's the singular for meninge. Right? And then you have the subarachnoid space, the pia mater, and the cortex. Okay. So your brain is very protected using all these different characteristics. And there's another way that the brain is protected, but this one is a chemical protection. It's called the blood-brain barrier. The blood-brain barrier means that the blood vessels in the brain are that the molecular structure of those veins are very tightly packed. Okay, so the blood vessels here have very, very tightly packed cells so that only very small molecules can get through the blood. Okay, so for the most part, what you're looking at is in the central nervous system. If you want a drug to interact on the brain itself, on the conduction of neurons and through neurophysiology, if you want that to occur, you're going to have to create a drug that is primarily lipid soluble and very, very small in molecular structure. And then to end this part, the physical protection, again, skull, meninges, and then cerebral spinal fluid. Okay. Let's talk about the neurons. Okay. Neurons are specialized cells for reception, conduction, and transmission of electrochemical signals. Neurons are like any other cell in the body. They have mitochondria, they have a nucleus, they have all that. But what is very special about a neuron is that they can communicate with one another. They come in many. Okay, sorry about that. I skipped some of the slides, and that was not the right thing to do. We'll talk about those different types of neurons in just a minute. Let's look at figure 3.5 here. And go through some of the terminology. You have the cell membrane. Remember I mentioned to you that it was very densely packed, so it's semi-permeable, which means that it allows some things to get through, but others not. And we just studied that primarily those things that will get through are lipid soluble and very small. Dendrites receive information from other neurons. And here's something to remember throughout the entire course, is that I try to provide you very clear and concise definitions for each one of these terms. But please realize that it is not as simple and straightforward as I'm making it out to be. I can say dendrite, and I can say, yeah, it receives most of the synaptic contacts from other neurons, but dendrites also transmit neurotransmitter. And so it makes it really confusing. Things can get out of control. So I'm going to try to keep it as simple as possible and give you very short definitions for things. I have done the same thing for the major brain areas. Okay, so we got the dendrites. Axon, axon hillock. Okay, this is the region at the junction between the axon and the cell body. So you can see there that that's just where the axon and cell body connect. You have the axon, which is primarily what is transmitting the neurotransmitter along there. Some neurons, or I should say, yeah, some axons are covered in myelin, which is that fatty insulation. When there is myelin around an axon, it will be white. Okay, so this is something that we'll get back to also. If you have a bundle of cell bodies, they look gray, so there you have gray matter. Okay? And where you have myelinated axons, it's going to look white. So your optic tracts throughout the brain are very white. The outer surface of the cortex, where you have cell bodies, is very gray. Okay? And then there's the cell body to the left of the picture there. Each segment of myelin is separated by nodes of Ranvier. And then you have the button-like endings of the axon branches. Some people call them axon terminals, axon buttons. And then you have the synapse. And one thing to remember about the synapse is that it really isn't a structure at all. The synapse itself is the space. It is just the space between a neuron and a dendrite, or I should say an axon and a dendrite. Okay? In this case, in this picture, doesn't always have to be that way. Could be an axon to an axon, could be an axon to a cell body. Okay, figure 3.6, the major internal features of a neuron. Everything in the top portion of this picture, you are not responsible for. I am not going to go over ribosomes, Golgi complexes, microtubules, any of that stuff. So don't worry about that for your quizzes. Don't worry about that for your exams. But at the very bottom, you have the synaptic vesicles and you have neurotransmitters. And all of this information is very, very necessary for Chapter 4. If you don't understand the basic anatomy, it's going to be really hard for you to understand the basic physiology. So the synaptic vesicles, those are the fluid-filled sacs. They actually contain neurotransmitter. Okay, and neurotransmitters are those molecules that pass from an axon to another neuron and influence that neuron in some way. And in Chapter 4, we'll discover what that is. And here's the cell membrane. Again, the lipid bilayer. The one thing to notice here is that you have the channel proteins, and those will become very, very important because that's how the molecules move in and out of a neuron. Okay. And then here's the slide that I was trying to get to before, but kind of messed up there. You have a unipolar neuron on the far left. You can see that it is just one solid pole. Okay. The second one, you see that there's a pole that extends from the top of the cell body, and then one that comes down from the bottom of the cell body. So now that's looking at a bipolar neuron. A multipolar neuron is one that has all kinds of dendrites, coming out of the cell body, and then one axon also coming out of that cell body. So it's a multipolar. And then finally you have the interneuron on the right-hand side, where there's no axon on that one. Okay, those are the four major types. Okay, glial cells. Glial cells don't get a lot of the attention. Definitely not like neurons do, but they play a very, very vital role in many different things. It says here glial communication and modulatory effects of glia on neuronal communication. So there's a lot of these things. They outnumber neurons 10 to 1. They work as a kind of a structure for neurons, but they have a lot of other purposes too. And let's look at the four classes here. So number one, you have the oligodendrocytes. Okay, oligodendrocytes are extensions rich in myelin, which means that it's going to look white, right? Because that myelin, that fatty tissue, is white. And they create the myelin sheaths for the CNS. Okay, remember CNS is going to be brain and spinal cord. So what kind of glial cells provide the myelin for the CNS? And that's going to be oligodendrocytes. Okay, so then what cells provide the myelin for the peripheral nervous system? And that answer is going to be the Schwann cells. Third on that list, you have the astrocytes. These are very large glial cells. They're astro, like astronomy, because they're star-shaped, and they have many functions. Some of those to actually respond to, as neurons die, it's the macrophage. They eat those dead cells, that dead neurons. They have those purposes. They also have a purpose of like scaffolding. So they hold neurons in place. Okay. And then finally you have the microglia, again micro, so they're going to be very small in this case, and they are involved in response to injury or disease, as I just mentioned, where they can actually it's like a cleaning mechanism. So a neuron dies, you don't want that tissue to be left there, so it's consumed by this microglia. Okay, couple diagrams here. So on the left-hand side, you see that big oligodendrocyte. That oligodendrocyte is providing, it looks like to me, 1, 2, 3, 4, 5, 6, 7, 8, 9, about 9 pieces of myelin. Okay, that is only going to be found in the central nervous system. On the right, you have a Schwann cell. Each Schwann cell is one myelin segment. It doesn't provide 9 myelin segments, it just provides 1. Okay, and those you're going to find in the peripheral nervous system. And we'll get into a chapter, I forget which one it is, where we talk about damage to the central nervous system, peripheral nervous system, and you'll have a better understanding of why in the peripheral nervous system we can regain function after nerves have been cut, but in the central nervous system it's very, very hard to do that. Okay, this kind of just all puts it together for you. Let's go to the second row down, clusters of cell bodies. All right, so you have in the central nervous system, you call them nuclei, and then the peripheral nervous system, we call them ganglia, clusters of cell bodies. Bundles of axons. In the central nervous system, we call those tracts. In the peripheral nervous system, we call them nerves. Okay. So remember, bundles of axons are going to be white. So tracts would be white, nerves would be white, clusters of cell bodies would be gray, central nervous system nuclei, peripheral nervous system ganglia. Okay, let's talk for a minute about ways in which we can view neurons, and that would be through neuroanatomical techniques, like the Golgi stain. The Golgi stain allows for visualization of individual neurons. Nissl stains selectively stain cell bodies. And we'll see some pictures in a minute. And then the electron microscopy yeah, provides information about the details of neuronal structure. There's lots of other ways of viewing the brain, and we'll talk about that in a later chapter also. But here, what we're looking at is just some of the staining techniques and also through electron microscopes. Okay. Figure 3.11 here. So this is a Golgi stain. Remember what Golgi stains do is stain the entire neuron. So you're actually looking at several neurons here. You can see where the cell bodies are. Kind of hard to tell where the dendrites would be versus where the axons are. Might be also kind of hard to tell whether it's multipolar, unipolar, bipolar, but nevertheless, you can see how some of the neurons were stained. In figure 3.12, now you're looking at a Nissl stain. And this would have been, these were the types of stains that I would use in the physio lab when I was doing this kind of research. And we actually tried to count these little dots in an area because what it's doing is it's staining the cell bodies. And so we would identify, in our cases, nucleus accumbens. We would look in the nucleus accumbens and identify where did this certain chemical concentrate. And we'd put a little grid down and then we'd count these. Nowadays, the microscopes are much more technical, and they actually shine a little grid down onto the slide of the neural tissue, and it takes a little picture, and what it does is it identifies if there's 300 neurons in this one little segment of this divided chart, then it does the math and tells you, okay, for that one little section of the slice, that's how many neurons there are. I wish I had that when I was doing this, but basically it's the same idea. Just technology makes it easier for us. Figure 3.13. Now what you're looking at here is electron microscopy. And you're identifying these are axon terminals, or what we said, those buttons. And the buttons here are very close to another neuron. It's hard to tell. This could be, again, a cell body, could be, or a dendrite. But now, remember what we were looking at just a minute ago was one multipolar neuron. And it had just this one long axon, and then it showed it connecting to another neuron. But in the brain, you can see how how busy it really is. There's hundreds and hundreds of axon buttons influencing this other neuron. And you might think to yourself, well, I've never mentioned that there's these axon terminals that are touching, or these axon terminals that integrate, or I try to avoid those terms, that terminology, because remember, between the axon buttons and the other neuron, whether it's a dendrite or a cell body, is a synapse, which means that there's a space between the two. Okay? So they're actually not rubbing against each other. Okay. Oh yeah, there's one other thing here for the tracing techniques. You have anterograde tracing and then retrograde tracing. Okay. And it's important to know that anterograde methods are used when an investigator wants to trace the paths of axons projecting away from cell bodies located in a particular area. Okay. And then retrograde tracing are used when an investigator wants to trace the paths of axons projecting into a particular area. Okay, so you might introduce some kind of a chemical into the cell body and find out where it goes, where it leaves that cell body, runs down the axon, and it goes. Anterograde, you might want to introduce a chemical into the space around that neuron, the extracellular fluid around that neuron, and see how it might be absorbed from the axon and then sent back to the cell body. So that would be retrograde backwards. Okay. Um, this is a section that is actually very, very important. These different directions, because now from now on, almost every single page in this book will probably have one of these terms used in it, because they want you to identify where these different brain structures are. Okay, even if 4, 5, 6 chapters from now we're talking about sleep disorders or something, then we're talking about the pons. They're going to talk about the pons, and they're going to say that those pons are ventral to something, or they're inferior to something. Okay? So it's very important that you memorize the directions now. It will make it much easier for you into the future. All right, so let's look at the figure on the left-hand side. Just remember that medial means middle. So medial towards the middle, lateral to the outside. Okay, so you could say that your ribs are lateral to your heart, and your heart is medial to your ribs. Okay, let's look at the cat on the right-hand side. So dorsal, most people don't have a problem with dorsal when they're looking at an animal, because you can remember dorsal fin on a shark or a fish. So that's the back. Okay, anterior, pretty easy, front, posterior, back. Okay, and then ventral would be down, or the stomach. So dorsal up, ventral down, anterior towards the front, posterior towards the back. Okay, sorry about that. I accidentally touched it again. Okay, so and then the one on the right hand side, medial, lateral, that makes more sense. Medial again towards the middle, lateral to the outside. Here's one thing that I think it's in the next slide, but what a lot of anatomists have done is that they've identified that instead of dorsal ventral, we just use superior inferior. Okay, so when we're talking about top of the brain superior, bottom of the brain inferior, and then you have anterior posterior. They still use anterior, ventral, dorsal, posterior for other parts of the body, but for the brain, to reduce the confusion with dorsal, ventral, we can use superior, inferior. Okay, and that's what this basically says right here. There's two other terms that are on this one here, though, that we didn't look at a minute ago, and that's going to be proximal. So proximal is close, and distal is far. So the terminal buttons are distal from the cell body. The axon hillock would be proximal to the cell body. Okay. Also, the sectioning of the brain is important for us. You can think of a horizontal plane as something that moves across, horizontal, a slice parallel to the ground. Okay, a frontal or a coronal plane or slice would be sliced down, like slicing a piece of bread. Okay, and sagittal is also a slice down. Okay, and it doesn't hit, but the sagittal is going to be running anterior posterior. So like on your PowerPoint slide here, it says a mid-sagittal section separates the left and right halves. Okay? But that just happens to be the mid-sagittal section. You can have other sagittal sections. They're going to be vertical. They're going to be slicing down just like a frontal, but they run from anterior to posterior. Okay? Which is different from frontal, which is also a slice downward, but it's running from side to side of the brain. This gives you a little bit better idea here. Okay, so this just happens to be a mid-sagittal because it's separating the two hemispheres, but it could be moved to the left, it could be moved to the right. Okay, but it's running from anterior posterior, whereas the frontal plane is running from from side to side, from the lateral most parts of the brain. Horizontal is a cross slice, and then you have a cross section also, which happens in other areas of the brain, or I should say outside of the outside of the cerebral cortex here. This is when you're sectioning like a piece of the spinal cord. If you cut a finger, you would do a cross section of the finger. Okay. Okay. A little bit on the spinal cord, not much gray matter. Again, cell body is white matter. That's because of the myelinated axons. The dorsal, okay, so dorsal top are primarily sensory in nature. They're afferent, which means they're coming into the central nervous system. Ventral, which means they're down to the bottom, more towards the bottom of the spinal cord. Efferent means they're exiting the central nervous system, and they're primarily used for motor, for moving muscles. Okay. We are getting through this material. There is a lot of material in chapter 3. Okay. Now this is the neural development. Here you're looking at what they call the neural tube. This is very early on in the development of the brain. So you have a spinal cord at the bottom, hindbrain, midbrain, forebrain. Then that splits up into the 5 major divisions of the brain. So you have the telencephalon, diencephalon, mesencephalon, metencephalon, and then finally the myelencephalon. Okay, and then the spinal cord. And we will be going through all of the major components. I shouldn't say all. We are going to go through the primary components. We leave a lot of stuff out. After this lecture, you're not going to believe me. You're going to think, man, we went over every single thing in the entire brain, but that's definitely not true. We are barely skimming the surface of neuroanatomy. Okay. Now, how do you memorize these things? The telencephalon, think of the top. Telencephalon is the top of the brain, and then as you move down, they're all in alphabetical order. So you have diencephalon, mesencephalon, metencephalon, and myelencephalon. Okay. Let's start on these. Okay. Here's another thing. It's kind of confusing. People kind of think from top to bottom when it comes from the divisions of the brain. But when you're dealing with the telencephalon, you can see how the telencephalon not only isn't on the top, but it encases, it covers up the diencephalon, the mesencephalon, and pretty much half of the metencephalon is covered up by the telencephalon. Okay? But those are your 5 different divisions there. All right, so let's start at the bottom. The myelencephalon, also referred to as the medulla, it's composed largely of tracts. Remember, tracts are going to be those, myelinated axons from the central nervous system. Origin of the reticular formation, something to think about here. The reticular formation actually runs through the myelencephalon, metencephalon, and mesencephalon. So if you read something, they say, well, the reticular formation in the mesencephalon, you say, well, it wasn't in the mesencephalon. Yes, it is. It actually runs through all 3. Okay. So the reticular formation is what you have for the myelencephalon. And like I said, some people refer to that just as the medulla. The metencephalon, here you have the pons and you have the cerebellum. The pons has a lot of different attributes here with sleep, respiration, swallowing, bladder control, hearing, equilibrium, taste, eye movement, posture, a lot of different things. Okay. Cerebellum, sensory motor, and coordination. And again, this is just to remind you again, I'm giving you a very, very clear and concise definition for each one of these. You can pick up journal articles, pick up another biopsychology book, and you'll say, well, wait a minute here, I didn't know that the pons did this. I didn't know the cerebellum did this. There are many other functions to these brain areas than what I'm just providing you in this lecture. The important part is that chapter 3, chapter 4, what they are doing is they are preparing you for all of the other chapters, so that when we talk about sleep, or we talk about memory, or we talk about hunger and so on, we can reflect back to the neuroanatomy and the neurophysiology, and we have a basis for trying to understand that material. Okay, so here what you're looking at is the medulla. So you have the myelencephalon in that light blue. In the purple, you're going to have the metencephalon. And then in that pinkish color, you're going to have the mesencephalon. And you see how the reticular formation runs through the myelencephalon, metencephalon, and mesencephalon. Okay, more on the mesencephalon. Now here what we're looking at is primarily the tectum and the tegmentum. And the tectum has the inferior and superior colliculi. Okay, the inferior colliculi is specialized for audition. The superior colliculi is specific for vision. The tegmentum has the paraaqueductal gray, which plays a role in analgesia. It also has the substantia nigra and the red nucleus. Okay, red nucleus because it is red, substantia nigra because it is black in nature, and those are both sensory motor. And that is the tectum and tegmentum. Then we move to diencephalon. Here's where you have the thalamus, and I refer to the thalamus as like a switching station because it takes that sensory information from the body and then it sends it. It's like a switching station. It says, oh, this is a pain attribute, and it needs to go to the somatosensory cortex in the parietal lobe. Okay. Oh, this is a movement thing that this individual wants to move their foot or something like that. The sensation comes up, goes to the motor cortex in the frontal lobe and says, move the foot. Okay. So it's like a switching station. We'll go into a lot more detail in a minute. The hypothalamus is all about eating, sleeping, and sexual behavior. And we will talk much, much more about the hypothalamus also. Here's a diagram of those areas so that you can see. You see the superior and inferior colliculi, part of the tectum. Then you have the tegmentum, which is just right below it. You're looking at the red nucleus. You see how it's red in nature. Substantia nigra is black in nature. You see the paraaqueductal gray, which is gray in nature. So those are the major components of the mesencephalon. And then the diencephalon, that's where you had the thalamus and the hypothalamus that we just talked about. Okay. Also, a couple other features that they bring into play here is the optic chiasm. The optic chiasm is where the optic tracts actually cross over. We'll talk more about that in the visions chapter. The pituitary gland releases hormones throughout the body, and then the mammillary body is responsible for, it actually plays a role in memory and specifically for spatial memory. We don't go into a lot of detail on the mammillary body. Some of these, the very few of these, I think that the author's done that on purpose. Very few of the terms do we not see again in a future chapter. I would say probably 95% of everything. But every once in a while there's one or two that we just don't focus on very much. All right, now check that out. Now we're up at the top. Okay, the telencephalon, or the cerebral cortex. The cerebral cortex is very convoluted. That means it's got all kinds of, it's really wrinkly. Okay, and that's so that more of the brain tissue can fit inside our skull. If it was all laid out flat, our heads would be ginormous. So it increases surface area there. A couple terms, and I don't know if your book does a very good job of these, so I put them in here for you. A gyri are hills, okay? So a gyrus is a small hill. A gyri is small hills, plural. Sulcus is a small valley or a crevice, however you want to think about it, small valley. So sulci are valleys, and then fissures are large valleys. A fissure would be singular, and fissures would be plural. Okay? So longitudinal fissure, okay? That's a groove that separates right and left hemispheres. So the longitudinal fissure would separate the right and left hemisphere. It's a very, very large valley. Okay? The corpus callosum is the largest hemisphere connecting tract. So there are other hemisphere connecting tracts, but the corpus callosum is the main one that connects the left and right hemispheres. Okay, and now we're able to see a lot of different brain areas in this slide. And I'm actually going to go through all of these brain areas one more time before the lecture is over. I've actually written them all out and defined them all myself, and we're just going to go right down the list. So instead of bringing up central fissure and lateral fissure and so on right now, which you can see in the chart here, I'm going to go ahead and define those in a little bit. Okay, but you do have so here you have the precentral gyrus. Okay, so that's a little hill that's in front of the central fissure. Then you have the post-central gyrus, which is again a little hill, but this time it's behind the central fissure. So the central fissure, as you can see right there, separates the frontal and parietal lobes. The lateral fissure separates the frontal and parietal lobes from the temporal lobe. Okay? So light blue is frontal lobe, purple parietal lobe, green is occipital lobe, and then the orange is temporal lobe. And I'll talk about all of those four lobes and stuff in a little bit also. Okay, the limbic system, part of the telencephalon. Here's where you have the mammillary bodies, the hippocampus, the amygdala, fornix, cingulate, septum. All of these, think about emotions and memory. We will definitely go into more detail on them, but think of them as a group. Limbic system, motor, memory, emotions. You're pretty good. Basal ganglia, you think motor system. Again, amygdala, striatum, which includes the caudate nucleus and putamen. And then globus pallidus also. So all of those parts are vital for our motor system. Basal ganglia in the telencephalon. Here's another graphic for you. So you can see the hippocampus, you can see the amygdala, mammillary bodies, septum. There's a term there that's a key one. I'm not too sure. Septum and fornix. Those are those things that they always list in every neuroanatomy diagram that you see, even intro to neuroanatomy or biopsychology, you see them, but they don't talk much about them in subsequent chapters. So those are a couple of examples for you. Okay. And this is just another graphic showing you some of the similar things. You have the putamen here, the globus pallidus here, in a little bit more detail. The nucleus accumbens also is provided here, and I mentioned that in actually the research that I did in the physio lab. And then we're going to end with this slide, but we're going to take some time here because for two reasons. I want to make sure that you have a basic function for each one of these neuroanatomical structures. Okay, because since this isn't a lab class per se, you're not dissecting the brain. I'm not providing you pictures of the brain, and you have to identify the different parts and that kind of thing. So it's more important in this class that you have a basic understanding of at least one function for each of these cortical areas. Okay, so that's my goal in this. So let's look at the telencephalon. And let's look at the cerebral cortex. Oh, the other major part to this also is, again, just for you to hear the same words over again. So when we say hippocampus, you can follow along and it'll make it easier for you. It just familiarizes yourself with the word, and it doesn't seem like it's such an alien concept for you. You'll be more comfortable with them. Another thing is that if you, at this time, if you want to take out a piece of paper and you want to write these down, or you could even write them in your book, however you want, I will go slowly through each one of these brain areas, and you can put that vital function on there. It'd be really helpful for you to study, because basically it's a summary of the entire chapter. Okay? At least for the cortical structures. I'm not going to do this for the neuron, but okay, for the gross anatomy, it would be. Okay, so let's get started. Cerebral cortex. First thing under that is the neocortex. The neocortex is 90% of the cerebral cortex, and it has 6 layers to it. Okay? And you're going to tell me a lot more about the neocortex in your e-lecture topic item. Okay, the second thing there is hippocampus. Hippocampus, memory, especially spatial memory. Right? How about the major fissures? Central fissure separates the frontal and parietal lobes. It's important, again, for you to know where the central fissure is so that you know where the pre- and post-central gyri are, because the pre-central is motor and the post-central is somatosensory. We'll talk a lot about those. The lateral fissure separates the frontal and parietal lobes from the temporal lobe. The superior temporal gyrus is specific for audition. Okay? And then you have the longitudinal fissure, which separates the left and right hemispheres. Okay? The major gyri, pre-central gyrus, just in front of the central fissure, which we just talked about. So just in front, central of the central fissure is the pre-central gyrus. Motor response, that is the motor area. The post-central gyrus is somatosensory. The superior temporal gyrus is audition and language. The cingulate gyrus is actually part of the limbic system, and you have emotions tied with that one. Okay, the 4 lobes. You have the frontal lobe, motor and complex cognitive functions. There's a lot to that. You can read more on page 69 of your book, but motor and complex cognitive functioning. Temporal lobe, audition and language. Parietal lobe, body sensations. Occipital lobe, vision. Okay? And then we're on to the limbic system. The amygdala, emotions, particularly fear. The hippocampus, memory, especially spatial memory. And then fornix, also memory. The cingulate cortex, the septum, and the mammillary bodies, you can combine all those together with emotions. And again, like I said, we don't go into much detail with those. Basal ganglia, again, amygdala. We already mentioned emotions, particularly fear. Then you have the caudate and putamen, which are known as the striatum. Why do you think it's called striatum? Because it's striped in nature. Okay. And then globus pallidus. And I would like to add, since your author doesn't talk about it in the text, but it's on one of those graphs, on one of the charts, the nucleus accumbens. Okay? Because all of these, striatum, globus pallidus, and nucleus accumbens, have an influence on voluntary motor response. Remember basal ganglia? A lot about motor there. But the nucleus accumbens has a special attribute with rewarding of, or the rewarding effect of drugs. The rewarding effect of drugs. So we'll get back to that one in a later chapter also. And then the cerebral commissures, the largest of those is going to be the corpus callosum, which connects the two hemispheres. And that is the telencephalon. And we move on to the diencephalon, where we have the thalamus. Okay, remember I referred to it as a switching station. The massa intermedia just connects the two lobes of the thalamus. The lateral geniculate nuclei is specific for vision. The medial geniculate nuclei, audition. And then the ventral geniculate nuclei, somatosensory or somatosensation. Okay. The hypothalamus, eating, sleeping, and sexual behavior is what it's specific for. Eating, sleeping, and sexual behavior. Mammillary bodies, again, memory, spatial. The optic chiasm, this is where the optic nerves come together and cross over. And then the pituitary gland, and they release hormones. Okay. And that's going to be the diencephalon. On to the mesencephalon. We have the tectum, which includes superior colliculi, that's for vision and visual motor response. Inferior colliculi, and that's for audition or auditory responses. And then we have tegmentum. And in the tegmentum, we have the reticular formation, and it's all about arousal. You can read a lot more about that on page 65, but we will go into a lot of detail on reticular formation too when we talk about hunger, we talk about sleep patterns, and those kinds of things. The cerebral aqueduct connects the third and fourth ventricles. The paraaqueductal gray has a role in analgesia. And then the substantia nigra and the red nucleus for sensory motor functions. A lot of different areas in the brain are devoted for motor movement, definitely. Okay. And that is our mesencephalon. We go on to the metencephalon. Again, reticular formation. Remember I mentioned to you, it passes through all three areas. Pons, specific for sleep, respiration, taste, posture. Again, a lot of different areas for pons. Excuse me, I had to sneeze there. Okay. And then the cerebellum, page 66, more information there, but sensory motor and cognition. We'll talk a lot about cerebellum. And then finally, well, that's the metencephalon. And then finally, there's the myelencephalon, and we have reticular formation. And we've already talked a lot about reticular formation since it's in three different areas. So that pretty much does it for our e-lecture on chapter number 3. And I look forward to doing this again for next week on chapter 4, where we can go over not only just the neuroanatomy, but then how do these different areas communicate with one another and how do they function? And that will be the goal of neurophysiology. Okay, thank you.